right on your screen. All right. In this context, you know that we will be having uh, we'll be having a DNA chat window also inside your app. We have a chat window. From the chat window, you are able to send messages to me, which I'll be getting in real time, and I'll be taking those messages also in real time. Okay, so please make use of that chat window. Keep sending your doubts and queries or any part you want me to repeat, so that we can interact in live time. The main benefit of this live session is from the comfort of your home with all the good food cooked by your mamas and sisters, you can simultaneously have the fun of taking the class. So this is for the first time that you can have the best of both worlds. You are at home, you are enjoying the luxury of the cooking of your parents and your siblings, and at the same time you can claim that I'm studying. So that is the biggest advantage of this session. We are having live classes, it's not a recorded session. So just like in class, if you have a doubt, if you have a query, you can send across your queries and doubts. If you want a topic to be repeated, if it is fast for you, then you can request that also. Students are joining us from all over the country. Some of them are even from outside the country. We have a few students from Nepal and from outside also. One or two students are joining from Kyrgyzstan, another couple from the Crimea Medical College. So this is you can say multinational platform, and that's why we have chosen one language, English. So we will be conversing in English only, because it is acceptable to all the different students that we have. We sometimes in different sessions we can have localized classes also, where we sometimes take classes in Hindi or some other local languages, but in these sessions we will be using only the English language, keeping in mind the wide variety and regions from which the students are joining us. All right. Oh, so a very brief overview of the sessions that we'll be having. We'll try to have 2 to 3 sessions, sometimes 4 sessions a week. At least 2 sessions for biochemistry will be there, and it is planned that 2 sessions each for the other two subjects will also be there for you. We are planning and trying to keep it in late evening, so that even when the lockdown ends, you can continue attending these sessions along with your college attendance, because as you can see, the timings are typically from 8 to 10 p.m., and by this time you can definitely make out 1 hours, 1.5 hour or 2 hours to attend these sessions. All right, so like I said, we'll be having a 2 day session for each of the subject, for anatomy, physiology and biochemistry. And the syllabus will be given to you, what is the schedule that we are going to follow. The biggest advantage of these sessions will be that your whole syllabus will be covered by one single excellent level faculty, which is generally an international level faculty. Throughout the theory part, the practical part, the viva voce, how to attempt the paper, what is the horizontal integration, what is the vertical integration, where you should know the clinical aspect, where you should know the topics from the other subjects from the same year. All of this will be covered by a single top-notch faculty. Now this is something which we as medical students used to feel when we're in the medical college. Some of the faculty are very good, they are top-notch, but it is not true for all the faculty. And because of that, there are sections of a subject where we lose interest, there are sections of the subject where we are not able to imbibe the most important knowledge or the information. So that part will not come into picture here, because one single faculty which has been selected from the best available for the classes has been appointed to take all your the complete syllabus for your class. Okay, so that's one. Second, like I said, in the new curriculum, there are two very important aspects where we have a common word: integration. And now, the Medical Council of India wants us to emphasize about the overlapping nature of the medical education, and they want us to emphasize to you that don't look at the subjects in isolation. When you are reading bio, which is working with the physiology, how I can use this knowledge that I am gaining in case of pharmacology, how to understand the action of the drugs using the biochemistry, how I can apply it into the clinics when I talk about the normal values of the enzymes, how they are going to be helpful to me when I see the patients, when I'm managing the patient, when I order the investigations, when I get the reports. So what is the aspect that I have to keep in mind? So those thought process should start coming from the first year itself. So that is one very important aspect. We call it the horizontal integration. Horizontal integration means when we tell you about overlapping aspects of the different subject in the same year. For example, when I'm teaching biochemistry, sometimes, let's say, when I'm talking about the acid base balance, I'll talk about the regulation of acid base at the level of kidney. So that is an domain of physiology. So that is an horizontal integration. When I talk about enzymes, I'll talk about enzyme inhibition, and at the same time I'll talk about how this enzyme inhibition is used by drugs for treatment. So that is a vertical integration. I'm integrating the biochemistry with the pharmacology. At the same time, I can talk about the enzyme levels in a particular disease. For example, when there is liver damage, what is the changes in the enzymes of the liver function? That is the alanine transaminase, the aspartate transaminase. So that is again a vertical integration with the clinical subject. Maybe we are talking about medicine, maybe we are talking about the pediatrics, maybe we are talking about the surgery. So the horizontal and vertical integration is one thing that we will continuously be talking about. I'll again repeat to those students who have joined us late. Inside your app, you have a chat section at the end. In the app, you have the chat section, and please make use of that chat section if you want to ask anything, if you have some doubts, if you want us to repeat some part. Uh, for starters, you can just send a hello or a hi so that I know that you have been able to see the chat section, you are able to encounter the chat section, and you are able to use it. So just send in your highs or thumbs up into the chat section, which will give us an idea that okay, you are there online on the system, and we get to know from where all the students have been able to join, or if somebody is having any issues with joining. You can also give us the feedback for the video feed, for the audio feed, and whether you are able to see the screen clearly or not. These are some things which you can tell us. If you are not still not getting it, I'll quickly show you in the app where you can see the chat section. So this is your DNA app, and at the bottom, right at the bottom, here we have the chat section. In the chat section, the MBBS course will be the chat section. Okay, great. So as you can see, there are already a lot of students with us. All right. So great, very nice to see all of you coming into our system. Very briefly, so that because now I can see a lot of students are here, I'll tell you a little bit about myself. I am Dr. Nilesh Chandra. I have done my MBBS from Maulana Azad Medical College, Delhi, and I have done my MD from University College of Medical Sciences, Delhi. I have been teaching students for last 8 years, almost 9 now, and I have been teaching at large number of coaching institutes. I have worked at the Subharti Medical College in Meerut as assistant professor. I have worked as senior resident as the University College of Medical Sciences and GTB Hospital. I have been visiting faculty at medical colleges in Kyrgyzstan, Crimea, Russia, and China, where I have taken classes for all students, whether they are in the first year or students who are preparing for the foreign medical graduate exam, who want to come back to India. And uh, Dr. Neeraj has given me this opportunity to come to you live from his platform, so that again, sitting from the comfort of our homes, we can interact with each other not only within the country but all over the world. Like I said in the beginning, we have various students joining us from Kyrgyzstan, some from Nepal, Crimea, and I think one student is there from China also, from Jiangsu, I think Jiangsu Medical College. I'm not sure. So this is a truly international platform, and it has been made possible with the efforts of Dr. Neeraj. And I would like to once again thank Dr. Neeraj for bringing this DNA app platform where all of us can interact together. Okay, so a little bit about the biochemistry section. I'll tell you what we are going to do and how we are going to approach the subject. Now, uh, biochemistry should look familiar to most of you because there is a very large overlap of topics. I would not say the content, but overlap of topics from your 11th and 12th. So in this sense, there is a comfort, a level of comfort with biochemistry, that okay, we are building on something which you already know, unlike in anatomy where it is almost very brand new, or physiology where we are going in too much depth of the superficial things that you had seen, along with a lot of new topics. Biochemistry, most of the topics are familiar. Obviously we are going into a lot of detail in those topics, and second part is, you'll note, it is focused. All the biochemistry is focused on the human biochemistry. Please note, now in the MBBS course, except for microbiology, all the time we'll be focusing on what is happening in the human body. All right, so when we talk about the cell, we'll generally not talk about photosynthesis, because in humans photosynthesis does not occur, whereas you talked about that in 11th and 12th. So that is one point to keep in mind. In our curriculum now, you have to focus on the human body, because the end vision should be clear. We are talking about treating, managing humans, so that part is very clear in our mind. We are going to talk about humans, and therefore all our discussion will be based accordingly. Okay, so like I said, uh, we will be talking about the theory part, how you are going to attempt the topics in terms of the question which are asked. I'll keep telling you where you should have sufficient content for a long question, where having a little bit of content for a short question, or differentiating between two topics, or just writing two lines, or the clinical aspect part is required. Because we have been seeing the papers over the last 8 9 years, how the questions are being asked at the university level. So we'll be able to discuss how much in depth we need to go, how much information we should know for a topic to be able to comfortably attempt it in the exam when it comes for the university exam. Now here I would like to tell two things which your seniors, immediate seniors, may not be able to guide you about properly. See, your immediate seniors are still MBBS students, and they might not know, or they might not have the complete picture of how this is going to progress, how the things are going to progress. So please understand that what you do and how you perform in your years, first year, second year, third year, fourth year, is going to have an impact on your career throughout. Some of your seniors might tell you that okay, it is important to pass because finally you're going to give the PG entrance exam. I would like to disagree with that opinion, because the first and second years are, number one, a very important fundamental, or you can say your foundation for the whole curriculum. If your first and second year knowledge is weak, you'll not be able to be comfortable and confident in your clinical subjects. If your anatomy is weak, surgery, orthopedics, maybe ENT will be very very shaky. If your physiology is weak, it will have a lot of difficulty. Even in pathology, you'll have a lot of difficulty in medicine, in pediatrics. So your foundation is very very important. If your biochemistry is weak, then the inborn errors, metabolism, genetic disorders, the various conditions related to nutrition, they are going to be weak. So your first year, second year are very important in building your foundations. So you must focus, put in extra effort and build strong foundation. Stronger is the foundation, the stronger and more stable will be the building. Number two, later on in your life, in your career, you will be going for jobs at multiple places. A few of us will venture out in private practice, but majority of us tend to find security in doing the jobs. When you go for the jobs, your whole curriculum vitae, your whole biodata is taken out. How do you do it in first year is the first thing which goes to the eyes of the you can say examiner or the person who's taking the interview. How much marks, what is the percentage in first year, that is the first thing which comes at the top. So please note, doing well in first year is critically important. Number one, it is important for a strong foundation. Number two, it is the first thing which meets the eye of your prospective employers later on when you go for the jobs. So please note, very, very important to build strong foundation, put in a little bit of extra effort in the first year so that you are much well off. You don't have regrets later on in your career that okay, if I would have done better in first year, maybe I would have been a little bit better off at this point of time. So I am giving this information to you because at this stage I have the whole picture of the career of a medical doctor. I have gone through MBS, I have gone through MD, I have worked as senior resident, I have worked as a faculty, I have worked as assistant professor, and I have been working with students at all levels, at the MD level, the post MD level, super specialty level, the people who are working in the private sector, people who are working in the medical college, people who are not working in a college but just in a government hospital. So I am working with all sorts of students at all levels of their career, and I have a good picture as a faculty that what as a you can say recruiter or employer I'll be looking at in a student's biodata. Okay, so that is one important thing I wanted to tell you. Yeah, okay, so now a little bit about the class. See, as you can see, you have a screen in front of you along with my picture. The screen is coming out to you from my iPad. On the iPad, I'll also be showing the PPTs where and as required. So I'll be writing, for example, writing here and saying hello to you. I'll be writing when I want to send across some information, or I'll be showing you some images, diagrams, tables. Also, I have some slides which I might choose to show to you. Probably in today's session, we will not be having a look at the slides and PPTs, but please note they will be there in the subsequent lectures and the sessions that we will be having. So that's one. Second, I'll be showing you lots of diagrams, particularly related to the practical work that you are expected to complete in your colleges. Please note, I'm repeating again, our sessions are not just the theory sessions. We'll also tell you how to tackle your practical work, what will be expected of you in your practical work. So that is another aspect that we'll be covering. It is a theory come practical session. We'll tell you how to prepare a subtopic or a subtopic for the theory exam. Will tell you what are the expected viva voce questions from a topic that we are discussing. Will tell you what is the vertical integration, the clinical aspect which will be asked. Will tell you how to write the notes, short notes, when you want to compare, when you have the question on compare and contrast. So we'll be approaching the class from the prospective MBBS end of year professional exam. We'll be preparing you to excel, to ace your exams at the end of the year, simultaneously building the strong foundations. In this context, very briefly, I would like to introduce the topic of biochemistry. About 30-40 years back, biochemistry was not a subject taught in MBBS curriculum. Before 1970s, we did not have a separate subject as biochemistry. It was a very small subsection within the physiology. In late 70s, it was started as a very small section. Biochemistry was started separately, and at that time we did not have any qualified MD doctors who had specialized in biochemistry. So mostly we recruited people from outside, and it started as a very small subject. but gradually it has acquired such huge dimensions that now we are talking about specialization and super specialization further in biochemistry. And biochemistry is a very, very important aspect in today's world of what we call evidence based medicine. In the subsequent years you will regularly hear this term, evidence based medicine. Very simply, whatever clinical decision a doctor takes today, it is simply based on the reports that the doctor gets. Obviously they do some amount of clinical examination which helps us to guide, to make a differential diagnosis what are the possible problems that an individual might be having. But which is the final diagnosis, the confirmation is based on evidence. The confirmation is based on evidence. And out of all this evidence, more than 85% evidence belongs to the topic of biochemistry. Majority of the blood tests that you will do fall under the section of biochemistry. So please understand, biochemistry has morphed into a very major subject. It has a lot of clinical applications. So not just for your first year knowledge, but for your future clinical practice, biochemistry is very, very, very important. Now, this is true for a large number of colleges, that a section of faculty in biochemistry is not from the medical field. And I personally feel that it is a major drawback, because for a person who is not from the medical field, it is very difficult to tell you or make you understand the importance of biochemistry later on in your career. So like I said, more than 85% of all the evidence required to manage a patient comes from the biochemistry. So it is very, very important to understand the fundamentals of biochemistry. What is the normal aspect of biochemistry? What are the normal parameters which you will find in an healthy individual? And that is where you need to understand the various sections in biochemistry. This is very, very important. All right, so biochemistry is the evidence-based medicine. Now, nowadays we have a very new section in biochemistry which has become very, very important, and that is the molecular biology. As gradually we are shifting from infectious disorders to inherited disorders, and even for the infectious disorders the molecular biology has come into play. So initially we started with the genetics. All of you know what is genetics. Genetics is the sequence of nucleotides in our body, and this gave us what is known as the genome. I'm giving you some new terms which you should understand, which is a part of the biochemistry picture. And we developed a section which deals with the study of genome. It is called epigenomics. It is called epigenomics. Okay, so the study of genome is epigenomics. Sorry, the study of genome is genomics. This was the first and important section which was there previously, but nowadays we have multiple new sections. From genetics we also started talking about epigenetics. These are some topics in which we'll go into the detail, and from here we get what is epigenome. Thank you. And the study of epigenome is epigenomics. So these are some new topics which you should keep in mind, that nowadays we are studying. Later on, it was discovered that a lot of other factors are related to causation of disease. Just genetics and epigenetics is not enough to understand the disease that is happening. And we started looking at the various macromolecules which are inside the cell. For example, we started looking at the carbohydrates which are present in the cell. We called it the glycome. Okay, so the study is glycomics. We started looking at the lipids. All right, we called it lipidome, and the study is lipidomics. Lastly, recently what we do, we give a stimulus, let's say a hormonal stimulus, let's say we add insulin or epinephrine to a tissue, and then we look at the response to the in the tissue. What are the metabolic changes that occur in that tissue? What are the different metabolites which are generated in response to a stimulus? And we call it the metabolomics. So what I'm trying to tell you is, you can see there is a lot of new knowledge, new information coming up in biochemistry, and all of this has a clinical application. These are some things which you should know. They will be asked from you when the examiner meets you, whether you know what is glycomics, what is metabolomics, what is epigenomics. These are new upcoming branches, or you can say subsections of biochemistry, which deal with particular segments of the biochemical feature of a cell. Previously we only had the genome, but we now talk of epigenome, the glycome, the lipidome, and the metabolites. At the same time, you should know about the basic composition of the cell. Basic composition of the cell, obviously you know this, you have covered this in 11th and 12th. So the basic composition of the cell in terms of what are the subcellular organelles. Now this is very very important. Knowing about the subcellular organelles is very very important. The reason being, different metabolic pathways are localized in a particular compartment in which organelle they are occurring. Certain proteins will be found only in a certain compartment. The metabolic pathways are limited to a certain compartment, and uh, the defect in a particular compartment is obviously going to affect the metabolic pathways which are occurring in those compartments. So we should know the different subcellular organelles and the metabolic pathways which are occurring in those subcellular organelles. This is very very important to understand, that and the cell and the subcellular organelles will help us to define and identify the various disorders. You already know that cell is identified as the building block of life. It is the building block of life. The subunits of the cell are not considered to have life. When they come together and they form the cell, that is when we consider the life to have appeared. You also know about the prokaryotes and eukaryotes. You have covered this in your 11th and 12th. So remember, in case of humans, we are mostly talking about the eukaryotes. Prokaryotes will discuss only in microbiology. When we are talking about infections, only then we'll be talking about prokaryotes. Otherwise, everywhere we are only talking about eukaryotes. Otherwise, we are only talking about eukaryotes. That is, we are talking about the cells in our body, the cells which are present in our body. I think the video is stuck. I think the video is stuck. Just give me 1 second. The video is stuck. Just give me 1 second. I'll restart the video section. Remove it. Add video capture device. Okay. Okay. Just bear with me for a minute. There has been loss of the video connectivity, which will come back in just a few seconds. Just a few seconds. Okay. Okay. Sorry for the loss of video. It happens sometimes. Technical problem, you understand. Okay. Okay. So like I said, in the human body we have the eukaryotic cells, and we will mostly be focusing on the eukaryotic cells. So very briefly I'll talk about some of the points that you have to know about the components of the eukaryotic cell. What are the different aspects that you have to consider, which will be a little bit a step up from what you have covered in your 11th and 12th levels. So first we said we have what are known as the subcellular organelles, and we want to identify each of the subcellular organelles so that we can study the different metabolic pathways occurring in these subcellular organelles. So the first question is how to separate these subcellular organelles. If you are not able to separate the subcellular organelles, then you will not be able to individually study the different metabolic pathways which are occurring in those subcellular organelles. So please note the first thing, the separation of the subcellular organelles is done by what is known as ultracentrifugation. I'm sure you know what is ultracentrifugation. When you carry out the centrifugation at a very high speed, it is called ultracentrifugation, and this ultracentrifugation is performed in multiples of g. You know what is g? G is the gravitational force. When you do the rotation, the centrifugal force is the g, and the force required here for separating the subcellular organelles ranges from 10,000 to 1 lakh G. After a short break, I'll show you the different speeds at which the different subcellular organelles can be separated. So, one very important step in studying the subcellular organelles and the metabolic pathways which are occurring there is the ultracentrifugation. By ultracentrifugation, we are able to separate the subcellular organelles into the individual fragments where a specific type of subcellular organelle is there. Okay, so you have separated it. Second is how to make sure that a particular fragment contains the particular cellular organelle that you want to study. Second is how to identify. Second question will be, once you have separated, how are you going to identify whether the particular fragment contains the subcellular organelle you want to study. So here we have what are known as markers. All right, for each of the subcellular organelles we have well defined specific markers which have been identified over the various decades of study. They have been identified, the specific markers have been identified which are present on a subcellular organelle. Sometimes you can have a single marker, sometimes there can be multiple markers which will help us to identify uh, the subcellular organelle. Okay, now we also have to understand that sometimes there can be overlap. When I say overlap, this means one pathway occurring in multiple compartments, means a part of the reactions occur in one compartment and the remaining part of the reactions occur in a second or maybe a third compartment. So we should also understand that there can be overlap of the pathways in the different compartments, and here you should be able to understand that only a part of metabolism will be visible in one compartment. When there is overlap, you defect in organelle is going to affect one or multiple pathways involving that organelle. See, it depends on the level of defect. If there is a single enzyme or a protein from the organelle which is missing, only a specific pathway may be affected. However, sometimes the organelle may be completely damaged, it may be absent, or it may be empty, there are no contents inside that. Then all the pathways which involve that organelle, all of them may be affected. All right, so this is another aspect which you have to understand, that if there is a defect in the organelle, then all the pathways involving that organelle may be affected. I'll give you an example. For example, we have a compartment called peroxisome. Now, you know that there are some specific metabolisms that occur in the peroxisome. For example, in peroxisome, there is oxidation of two types of fatty acid. Number one, very long chain fatty acid. There is metabolism of very long chain fatty acid. In addition, there is also the metabolism of the branched fatty acid. All right, there is the metabolism of the branched fatty acid. Now, when we talk about peroxisome, there are certain defects in the peroxisome, and because of that, we have two different types of disorders. One is the Refsum's disorder. In Refsum disease, if you examine the pathophysiology, a single enzyme from peroxisome is absent. Alright, because a single enzyme is absent, it is likely to affect a single pathway, and we find that in Refsum disease, the branched fatty acid metabolism is affected. The branched fatty acid metabolism is affected, and they will start accumulating in Refsum disease. Typically, this branched fatty acid happens to be the phytanic acid coming from plants in our diet. The phytanic acid that is coming from plants in our diet. But we have another disorder affecting the peroxisome, which is known as the Zellweger's syndrome. Another disorder, that is the Zellweger's syndrome. Now, in Zellweger's syndrome, what you find, the peroxisome is absent. Either it is absent or it doesn't have any content. It is absent or it is empty. So obviously in this case, the metabolism of the long chain fat, very long chain fatty acid, as well as the metabolism of branch fatty acid, both of them will get affected, because all the enzymes, all the metabolic pathways which involve the peroxisome will start getting affected. All right, so this is just to give you an overview of why we need to know these things. What is the importance of biochemistry? What is the new upcoming evolution in biochemistry? Like I said, biochemistry is continuously evolving more and more. Molecular biology, currently we all are passing through the COVID crisis. You know, we are sitting at home in the COVID crisis, and the diagnosis of this COVID virus is done by the reverse transcription PCR, which is also a part of the molecular biology, that is biochemistry. So the basis of RT-PCR is pure biochemistry, the molecular biology part, where we first convert the RNA into DNA and then we carry out the PCR. We will be talking about this technique when we do the molecular biology section. So what I'll do here, you can take a very short break. We had our introduction about what we are going to do and how we are going to do. You can have a cup of tea, you can have a plate of pakoras or a sandwich, you're sitting at home, and we come back in 10 minutes, and then I'll show you a few important slides, the distribution of the tissues, at what speed we do the ultracentrifugation for separation of the organelles, and those similar topics I'll quickly discuss with you, which will give you an overview of biochemistry, what you should be knowing about biochemistry going forward. All right, so we'll take a very short break, 10-minute break here. You can have a cup of tea. If you are sitting at home, you can go have a cup of coffee also. If you want, you can have a small bowl of Maggie or soup, whatever catches your fancy, and you come back in 10 minutes. Very short, you can say a tea break for 10 minutes. All right, so please be back in 10 minutes. हाँ प्रणाम, फोन कर भाजी। वो तो हम पूछ रहे थे कि माँ का क्या प्लान हुआ, कब जाने का, क्या करना है, कैसे करना है, उसके लिए पूछ रहे थे। अच्छा, तो बताइए क्या करना है। हाँ, बोले क्या करना है माँ का, अभी तो आज तो साइट क्रैश कर गया उसका। हाँ, अच्छा, करते हैं बात। अभी तो कभी तो यूज़ किए नहीं, हम तो ऑनलाइन ही निकालते थे, देखते हैं, पता करते हैं कुछ रखें तो निकल जाएगा। वाह, ठीक है, ठीक है। हाँ, आज तो सारा सिस्टम ही क्रैश हो रखा था आज का। हाँ, ठीक है, ठीक है। हेलो। नहीं, बस वही पूछ रहे थे। ठीक है। हाँ, वही 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 वही। हम्म, ठीक है, ओके। हम्म, अच्छा, अच्छा, अच्छा।
अच्छा, अभी तो सेशन माने बहुत ही अच्छा है। बच्चे कुछ लिख रहे हैं कि नहीं लिख रहे हैं?
एक सेकंड।